So I think we'll go ahead and get started. So today we are still about a week ahead of our lecture in terms of lab. But today we'll be focusing on gene expression. Okay, so for gene expression, this is going to be the process of being able to produce certain proteins based off of activating a certain gene. Okay? And so in order to perform gene expression, what we need to do is we need to go through a couple of processes called transcription and translation. Okay? So we're going to be looking at DNA. We're going to be looking at the full processes to how we're going to be looking at how our DNA is going to end up helping us to produce some proteins that will give us the characteristics that we're going to be able to express, whether it's physically or proteins that we're going to be expressing internally or within our cells, things like that. So that's going to be the goal. We're going to be looking at how we're going to be producing specific proteins. Okay. Now, if we remember when we first introduced DNA a few weeks ago, one of the things that we mentioned was that the DNA is going to have specific genes, and those genes are basically going to be the instruction manual to help us produce various types of proteins. Okay? And so we didn't really get into that much detail about it, but today we're going to be looking at that in a little bit of detail, and then we'll focus on it in lecture next week. Okay, so the whole goal of DNA is basically to act as an instruction manual to help us produce some proteins at the end of this process. Okay. The problem with DNA is it's a really big instruction manual. There's a lot of extra stuff in there that we don't need to help us produce this one singular protein. Okay. And so what we're going to be doing is we're going to be taking that DNA and we're going to be transcribing it into kind of a shortened instruction manual, essentially. Okay. That shortened instruction manual is going to be the RNA. Okay. And that RNA is going to be the short instruction manual that tells us exactly how we need to produce the protein. Okay. So this whole process is going to start with our DNA, which is very, very, very large instruction manual with a lot of extra information that we don't need. Okay. We're going to transcribe that into RNA, okay, specifically messenger RNA or mRNA. Okay. And then from there, that mRNA is going to be translated into some protein. Okay. So we're going to use that mRNA to help us produce the protein that we want to be producing at that point in time. Okay. So this is what's called the central dogma of biology. Pretty much everything in biology revolves around this one this one kind of trait, or this one pattern here. Okay. We're going to be starting with DNA. That DNA is going to be used to help us make RNA, and then from there, that RNA is going to help us make proteins. So this is a central theme in biology that's going to be really important going forward here. Okay. So when we look at transcription and translation, transcription is going to be a process that takes place inside of the nucleus. Okay. So here's going to be where we're going to find our DNA. The DNA is going to get transcribed into mRNA, specifically inside the nucleus here. Once we form our mRNA template, okay, the mRNA is going to get spit out of the nucleus, okay, and into the cytoplasm. So then it can now undergo translation to help us produce some protein here. Okay. So that's the goal for today. Okay. Now, before we dive into transcription and translation, we need to start at the beginning. Okay. We need to start with the DNA. Okay. And so when we're looking at DNA, okay, DNA is going to be found in a specific structure called a double helix. So if you remember from like high school biology, you've probably seen a DNA all wound up in a double helix formation. Okay. Now that double helix is going to have two strands of DNA. Okay, there's going to be one strand over here, okay, and then we're going to have a second strand over here. Okay. Now each of these two strands needs to be attached to the other one. Okay. And the way those two strands are going to be attached to each other are going to be by these nucleic acids. Okay, these nucleic base pairs are also called nucleotide bases. Okay, and these are going to attach one side of the DNA to the other strand of DNA. Okay, now if you kind of unwind that DNA and into more of a ladder formation, you're going to see that these nucleotide bases are essentially going to almost form like the rungs of your ladder. Okay, the stepping stools of your ladder here. But they're not going to be just linked to each other any which way. Okay? There's going to be a specific manner in which we're going to be forming these nucleotide base pairs. Okay? The first one that we're going to see is we're going to find our adenines or our A's. Okay? Those are always going to be attached to the T, the thymines, on the opposite strand of DNA. Okay? So A's will always bind with T's. So adenines will always bind with the thymines. Okay. On the flip side, we're also going to have our G's, our guanines. Okay. Our guanines are always going to pair with the cytosines on the opposite strand of DNA. Okay. So A's will always bind with T's, G's will always bind with C's. Okay. So we're going to keep that in mind going forward, because that's going to be a really big kind of important thing going forward here. Okay. So A's will always bind with T's, C's will always bind with G's. And so what that tells us is that the amount of adenines will always be equal to the amount of thymines, because they're always going to be in pairs with one another. Okay. And then the C's, our cytosines, will always be in the same amount as our G's, our guanines, because again, they're always going to be in pairs with one another. Okay. So the amount of A's is always going to be equal to the amount of T's, the amount of C's will always be equal to the amount of G's. Okay. And so we call these complementary base pairs because these are all nucleic acid base or nucleotide bases. Okay? These nucleotide bases are going to be complementary to each other, meaning that they're going to bind with one another to help hold the DNA intact with one another, okay? and able to hold it together. Okay? So this is going to be these base pairs that are going to hold our DNA together here. Okay? So we have a quick question here. So here we have a DNA sequence on one strand is as follows. Okay, so we have ATG, CAG, TA. So what will the complementary sequence on the opposite strand of DNA be? Okay. What do we think? So again, remember, A's will always go with T's and C's will always go with G's. Okay. So which of these would be the appropriate complementary strand? What do we think here? So I got two answers. One of them is correct. Yes, so C is the correct answer. So the opposite strand, the complementary strand of DNA is always going to be opposite to whatever you have on this strand. So here we have an A, so our first one is going to be a T. Here we have a T, so the opposite is going to be an A. Here, excuse me, we have a G, so we're going to have a C, C, so we're going to have a G, A, T, we're going to have a G here and a C here, T here, so we're going to have an A, and then an A, so we're going to have a T here. Okay, so this strand of DNA is going to be complementary to our original strand here. Okay, so this is going to be the complementary sequence, okay, on our DNA. Cool. Okay, any questions so far? Let's see if we can, and does this make sense? Okay. Okay, so if there's no questions, we'll go ahead and move forward here. And so that is going to bring us to our first process called transcription. Okay. So we looked at our DNA first, and so transcription is going to be the process of starting with the DNA and helping us form our messenger RNA or mRNA. Okay. Now in eukaryotes and prokaryotes, this process is going to take place in different locations. Okay. So in eukaryotes, transcription is going to take place inside the nucleus, okay, to form that mRNA. Okay. Once that mRNA gets produced, you're going to push it out of the nucleus and into the cytoplasm. Okay. In prokaryotes, because there is no nucleus, transcription will take place just inside the cytoplasm. Okay. And so when we take a look at transcription, transcription is going to happen in three phases. Okay. There's going to be an initiation stage, there's going to be an elongation stage, and then there's going to be a termination stage. Okay. So these are the three stages for transcription. Okay. So our first stage is the initiation stage. And so what we're going to do here is we're going to unwind the DNA double helix. Okay? So instead of it being kind of all helical, we're going to unwind it so it's more in its ladder formation here. Okay? So we're going to unwind our DNA, and then we're going to take a region of our DNA, okay? And you're going to pull apart those two regions, those two strands. Okay? And that's going to form what's called the transcription bubble. Okay? And so that's going to be this portion here where we see our strands being pulled a little bit further apart. Okay. This is going to get pulled further apart because this is going to create a little bit of room for us to be able to produce our RNA strand, okay, our mRNA strand. Okay. So In initiation, what's going to happen is we're going to start off with a bunch of proteins and a bunch of enzymes binding to a region on the DNA called the promoter. Okay. So the promoter is this region here. The promoter is always going to be just upstream of the gene that you want to transcribe, okay? The gene that you want to activate here. And so that is going to tell you where transcription is going to begin. Okay. So you're going to have all your proteins and enzymes attaching here at the promoter. Okay. That'll tell you where transcription is going to begin. And like I said, that promoter is usually going to be a little bit upstream on the strand of the gene you want to activate, the gene you want to transcribe. Okay. So that's all initiation is. We just unwind our DNA, okay, form our transcription bubble, and then have our enzymes and proteins binding onto the promoter region of the DNA. The next thing that's going to happen is we're going to start forming our mRNA using one of these strands of DNA. Okay? The strand of DNA that we're going to be using to help us produce the mRNA is called the template strand. Okay? So here we have one strand of DNA. This is our template strand in this case. The opposite strand is the non-template strand. Okay? So we're starting with our template strand here, and we're going to have a specific enzyme called RNA polymerase. And so what RNA polymerase will do is it is going to add some RNA nucleotides, okay, in a complementary manner to the template strand. Okay. And it's always going to be adding in the 5' to 3' direction. Okay. So here's what I mean by that. On our DNA, okay, each DNA strand is going to have a 5' region, which we'll talk about next week. And then the opposite end of that DNA strand is going to have the 3' region, okay, the 3' end. Okay. The opposite strand is going to have it in the opposite direction. So the 5' end would be across from the 3' end on that side. And then here's our 3' end over here. Okay. Our strand is going to have a similar situation going on. Okay. So it's going to be adding its RNA nucleotides, okay, from the 5' to 3' direction. So it's going to be adding those nucleotides onto the 3' end of the RNA. We'll talk more specifically about what that means next week in lecture. For now, just know 5' to 3' end. Okay. But like I said, the RNA nucleotides are going to be added in a complementary manner to the nucleotides on the template strand. Okay. So I'll kind of show you a little bit of an example on this next slide here. Okay. So let me go clear this out real quick. Okay. So here's our elongation phase here. So there is a slight caveat to where our elongation, or how we're going to produce our mRNA. Okay. So here we have our A that's going to bind to our T. So that's going to be our RNA strand. Okay. That's going to be the complementary base for this thymine. Okay. Now, when it comes to adding thymines inside RNA, okay, RNA does not have any T's. Okay. All those T's end up getting replaced with U's for uracil. Okay. So uracils will always take the place of T's inside RNA. Okay. So RNA, there's no T's. All we have are U's instead of T's. But everything else is going to be the same. You'll still have A's, you'll still have C's, you'll still have G's, but there will be a U instead of a T for RNA. Okay. So again, here we have our template strand. Okay. So here we have a T on the template strand. Its complementary base would be the A. So we have an A right here in the RNA. Okay. In the template strand here, we have an A. So its complementary base in RNA would be a U instead of a T. Okay. Here we have a C. So its complementary base would be a G for the RNA. Okay. Again, here we have a T in the template strand. So we're going to have an A in the RNA, and so on and so forth. Okay. So the mRNA that you produce is going to be built complementary to the template strand. Okay. Okay. So does that part make sense so far? Any questions on that? If this makes sense, go ahead and throw a check mark, or let me know it makes sense or something. Okay. All right. So that's the RNA in regards to the template strand. Okay. So the RNA is always going to be complementary to the template strand. Okay. Now, the other thing to keep in mind here is that the non-template strand of the DNA, okay, so this is the other DNA strand here. So I'll just put DNA. Okay. So the non-template strand, or also called the coding strand, is also going to be complementary to the template strand because they're opposite sides of the same DNA. Okay? So the coding strand is complementary to the template strand. The RNA is complementary to the template strand. So what that means is that the RNA and the non-template strand are going to be almost identical. Okay? The only difference here is that instead of having T's inside the RNA, there's going to be U's. Okay? But the RNA and the coding strand are basically going to be identical because they're both going to be complementary to the template strand. Okay? So that's one thing that you want to make sure that we keep straight. Okay, we want to always be focusing on the template strand when we're producing our RNA. Okay? But if you don't have the template strand and you do have the non-template strand, you can always use that to help form the RNA. You're just going to be identical. Okay? We'll worry about that more so next week. Okay, okay, so that's elongation. So we're going to be continuing to produce our RNA strands in this manner here. So actually, let's go ahead and continue adding to our RNA strand here. Okay. So here we have an A. So what would be the next nucleotide? U, right. Okay, perfect. Then here we have a G. So it would be the next nucleotide. C, perfect. Okay, then we have a C here. So what would we have? G, beautiful. Okay. Next we have an A, so we should have U, perfect. Okay. And then we have T, so we should have A, perfect. And the last one we have another T, so we should have an A, perfect. Right. So here we just built our entire RNA strand from our template strand. Okay. And then again, if you look at the non-template strand, we're going to see it's going to be basically identical. The only difference is that we're going to have U's instead of T's in the non-template strand, okay, compared to the RNA. Okay. This makes sense so far. Yeah, okay, cool. Okay. All right, so then, let me go ahead and clear this. Okay. So now we're going to continue finishing our elongation process. And so what's going to happen is the RNA nucleotides that we initially produce are going to be bound, okay, to the nucleotides on the template strand of DNA. Okay? They're going to be attached for a very short period of time. Okay? But then over a period of time, once the RNA polymerase continues to move, it's going to eventually release. Okay? So the RNA strand does not stay bound to the DNA. It eventually becomes its own individual structure. Okay? Now, as the DNA polymerase continues to move ahead, okay, the DNA is going to get unwound ahead of that DNA, okay, sorry, ahead of that RNA polymerase. And as RNA polymerase moves out of that particular region, the DNA behind it is going to get rewound back into that double helix. Okay? So you unwind it as RNA polymerase starts to enter that region, and then as it starts to exit, you start to rewind it back up. Okay? And you continue forming your RNA strand here in the 5' to 3' direction. Okay. Okay. So here we have a little bit of activity. So I'm going to go ahead and switch the screen over to, where is it? Okay. So this is the screen I want you guys to be looking at here. Okay. So is everyone able to see this activity screen here? Yeah, okay. Okay, so here, this is a little activity that says what we're going to be doing here is we're going to be transcribing and translating a particular gene. So here we're looking at our nucleus, and we've got a bunch of genes all located in here. So what we're going to do is we're going to choose one of these genes. Let's go ahead and pick this one. And we're going to try to transcribe this particular DNA sequence. Okay. Once this thing gets going here. So I'm going to go ahead and just kind of, okay. So give me one quick second. So, okay. So here we have our DNA sequence. And so now what we want to do is we want to insert the correct nucleotide into, that's going to be the correct base pair for this T. So what should be the correct base pair for forming our RNA? So we have a C, an A, G, or U. Right, we should have an A. Perfect. So we add our adenine. Okay, now we have a G. So what should we add here? C. Perfect. Okay. Then here we have a G, so we should add a C. Perfect. Okay. Here we have a T, so we need to add which nucleotide? A. Perfect. Okay. Then we have an A, so we need to add a what? U. Perfect. Okay. Now we have a T. So we should have A. Perfect. Okay. And then we have another A. So we need to add a U. Perfect. Now we have a G. C. Perfect. And then another G. So another C. Another G. So we have another C. Another G. So we have another C here, and another one, and another one. Perfect. So now we got a T, an A, perfect. Okay, and then another T, so we should have another A, perfect. Okay, and so now we've just formed our mRNA, okay, through this process of transcription. Okay, so I'm gonna go ahead and switch back to our PowerPoint, and we'll come back to this when we are ready for translation. Okay. Okay, is everyone able to see the PowerPoint again? Yeah, I know. Okay. Okay, is everyone able to see the PowerPoint now? Yeah, so it does say translation, yeah. Yeah, so we have just finished transcription through this process. So now, the next process that we're going to be doing is we're going to start with translation. Okay, so we finished transcription, we formed our mRNA. Okay, once we formed our mRNA, okay, now we're ready for the next process, which is translation. Okay, so we've taken that very large instruction manual in our DNA, we've condensed it down to a more concise instruction manual, which is our mRNA. Okay, and so the next thing that we need to do is we need to take that mRNA template. Okay, and then we need to use that to help us produce the protein. Okay, so that's what translation is. This is going to be where we're going to begin our protein synthesis by breaking down our mRNA message, by breaking down our mRNA sequence here. Okay, so again, in order to produce our proteins, okay, what we need to do is we need to link a bunch of amino acids together. So remember, each amino acid is going to be linked together to help us form a polypeptide and to help us form proteins. So there's going to be lots of covalent bonds that are going to be attaching all these amino acids to help us form our proteins here. Okay. And then from there, those proteins will be able to perform a variety of different roles and functions within the cell and then throughout the organism as a whole as well. Okay. That's translation. So before we get into how translation works, there's a few different components that are going to be important to help this process work. Okay. The first thing that we need is the mRNA template that we just produced through transcription. Okay, we're going to need some ribosomes. Okay, then we're going to need some another type of RNA called tRNA or transfer RNA. Okay, and then we're going to need some enzymes. Okay, so all four of these components are going to be necessary to help us produce a protein, okay, through translation. Okay, so before we get into that, which of the following are true about ribosomes? So there's two answers to this one. So we got A, we have ribosomes function in glucose synthesis, they function in nucleic acid synthesis, they function in protein synthesis, D, they're found on the smooth ER, E, they're found in the rough ER, and F, they're found on the Golgi apparatus. Which ones are true? And there's multiple answers for this one. So Brianna says E. So E is absolutely true. Yes, there's one other one that is also true here. C, yes. So C is correct. Okay. So C and E are both correct here. They're both going to be, ribosomes are going to be involved with protein synthesis. Okay. So they're going to be involved in helping us produce proteins, and they're going to be found on the rough endoplasmic reticulum. Okay. Okay, cool. All right, so starting with ribosomes. So ribosomes are made up of a different type of RNA, a third type of RNA called rRNA, or ribosomal RNA. Okay, so at this point, we've seen three different types of RNA. Okay, we've seen messenger RNA, we've seen rRNA, ribosomal RNA, and then later we're going to see transfer RNA, tRNA. Okay. So rRNA is a type of RNA that gets made inside of the nucleolus, okay, inside the nucleus here, and it's going to help produce ribosomes. Okay. So in prokaryotes, because prokaryotes do not have any organelles, okay, any membrane-bound organelles, the ribosomes are only going to be found floating around through the cytoplasm. In eukaryotes, ribosomes are going to be found on the rough ER. Okay. So these ribosomes are going to be made up of ribosomal RNA. That's one of the big keys here. Okay. And then for now, we're just going to focus on ribosomes being found on the rough ER. We're just going to be looking at eukaryotes for now. Okay. Now, each ribosome is going to have a small subunit and it's going to have a large subunit. Okay? The small subunit is going to be this part right here. Okay? The large subunit is going to be this more top part up here. So the small subunit's job is to bind onto the mRNA. Okay? The large subunit's job is to bind onto the tRNA, the transfer RNA. Okay? They initially are two separate components, okay? But when translation is ready to begin, the two subunits will then come together and then allow translation to begin. And we'll look at that in a second. Okay. So that's rRNA. The next type of, component to this process is called tRNA, or transfer RNA. Okay. Transfer RNA is really important to the process of translation because what it's going to be doing is it's going to be bringing in each specific amino acid, okay, to help us build that protein. Okay, so this is one full tRNA molecule here. Okay. And at the top of each tRNA molecule, there's going to be a region that's going to be bound onto a specific amino acid. Okay. And this amino acid will be used to help build the protein or the polypeptide sequence that we want. Okay. So we're going to add that amino acid to the growing polypeptide, and that will help us produce our protein. Okay. So the way this is going to work is our tRNA is going to bind to a specific region on the mRNA. Okay, so it's going to bind specifically to the mRNA template. Okay, and as it binds, it's going to be able to add that amino acid onto the growing polypeptide. And again, we'll look at what that looks like in a second. Okay. So this is what's going to be the actual component that actually translates the mRNA into an actual protein. Okay. Okay. That's that. Okay, so that's kind of the last player in this process. And again, we'll look at how this whole process will work in a second here. But before we get into the kind of machinery and the kind of mechanism of translation, there's a few things I want to kind of get straight first. Okay, number one, the one thing that we want to keep in mind here is that during transcription, we produced mRNA, okay, during transcription. Okay. During translation, what we're going to be doing is we're going to be taking the mRNA that we just produced from transcription, okay, and we're going to want to form a protein product using the mRNA that we had just made. Okay. And each of those proteins are going to be made up of amino acids. Okay. Now, in total, we have 20 naturally occurring amino acids in nature. Okay. And so any combination of those amino acids can be used to help form a specific protein. Okay. Sometimes you might have a protein that's 5 amino acids long. You might have some that are, 1 million amino acids long. But you're going to have any one of those 20, okay, all linked together to help form a very small or very large protein. And we'll look at that in a little detail here too. Okay. So how do we know which amino acid we need to add? Okay. That is going to be a key here. So the way we're going to be doing that is we're going to be looking at the mRNA sequence. So here's our mRNA here. Okay. In order to determine which amino acid we're going to be adding, we're going to be looking at each sequence of 3 nucleotides. Okay. So here we have 1, 2, 3 nucleotides here. Each group of 3 nucleotides on your mRNA is called a codon. Okay. So one codon is made up of 3 mRNA nucleotides. So this would be one codon, this would be a second codon, this GUC, this AGU would be a third codon, CCA would be a fourth codon, and so on and so forth. Okay. So each codon will code for a specific amino acid. Okay. And we'll look at that in a second. Okay. So each codon codes for a specific amino acid. 3 nucleotides make up one codon, and each codon codes for one amino acid. Okay. So the relationship between each codon and the amino acid that it corresponds to is called the genetic code. Okay. And so this is what the genetic code looks like here. Okay. So here is all the combinations of codons that we can possibly have. Okay. So we have 64 total combinations of codons, but again, remember, we're only going to have 20 naturally occurring amino acids. Okay. So there's going to be some repeats here. Okay, so for example, here we're looking at the amino acid arginine, ARG. So this amino acid is going to be coded by 1, 2, 3, 4, 5, 6 different codons, which are able to produce the amino acid arginine. Okay. Here we have 1, 2, 3, 4 different codons that can possibly code for one amino acid in threonine. Okay. So there are some repeats, okay. And there's some that don't code for any, and we'll look at those in a second here. Okay. So each codon is going to code for a specific amino acid. Okay. To begin translation, okay, there's a specific codon that we're going to be looking for. Okay. And that codon is called the start codon, and that is the AUG. Okay. So AUG is the first codon for any protein that we're going to be producing. That's going to be the first one where we're going to see translation occur. Okay. So this is a start codon, and this will also code for the amino acid methionine, or MET. Okay. So once you find the start codon, that's going to be the first one, and then you start looking for every 3 nucleotides to give you your next codon, and each codon will again code for a specific amino acid. Okay. Once you're done with translation, okay, the way you stop translation is you reach one of 3 stop codons. Okay. Those stop codons are either UAA, UAG, or UGA. Okay, those are the 3 possible stop codons we can end up with. Okay. And so the way you can kind of remember that is UAA for you are away, UAG for you are gone, UGA, you go away. Okay. Those are the 3 stop codons. Okay. The stop codons do not code for any amino acids. Okay. So whatever amino acid you had right before you hit the stop codon becomes the last amino acid of that protein. Okay. These stop codons just tell you when to clip your protein and when it starts to go off into the cytoplasm and do its thing. Okay. So these ones do not code for a specific amino acid. They just tell you when you're stopping translation, when you stop your protein synthesis. Okay. Okay. Any questions so far? Is this all kind of making sense here? This is making sense. Go ahead and throw a check mark or a heart or something and let me know. Sounds like science fiction. Yeah, it does sound like science fiction, but it's crazy because this stuff actually happens, which is pretty incredible. This is kind of where you know like biology is pretty dope. And how everything just kind of all works together in exactly the right way it's supposed to. So that's that. So we'll take a look at a few ways of how this is actually going to take place. Okay. Okay. So let me. Okay. So this slide here just kind of shows you what each of the amino acids full names are. Okay, so these ones on the right are the 3-letter abbreviations for each of the amino acids. Here are the full names of these amino acids. So alanine, arginine, asparagine, and just yada, yada, yada. Okay, so those are all the names of the amino acids. Okay, all right, so now we're going to take a look at translation in action here. Okay, so again, translation is going to take place in 3 stages. It's going to be an initiation stage, an elongation stage, and then a termination stage. And this is going to happen pretty much the same way in prokaryotes as it will in eukaryotes. Okay. So here's the way this is going to work. First phase that we're going to have is called the initiation phase. And so to start initiation, okay, what we're going to do is we're going to form what's called an initiation complex. Okay. That initiation complex is going to be made up of the small ribosomal subunit, okay, which is this guy over here. Okay. We're going to have our mRNA template that we produce from transcription. Okay. Then we're going to have some 3 initiation factors, which are just enzymes that are going to be involved with this process. We're not going to worry about what those are. And then there's going to be an initiator tRNA. Okay. This initiator tRNA is always going to bind onto the start codon, and it is always going to bind, which is specifically going to be the AUG, and it's going to be bringing the amino acid methionine, MET, okay, which we saw AUG is going to code for. Okay. So this would be the full translation initiation complex here. Okay. We have our small subunit, our mRNA, and our initiator tRNA, which contains our methionine amino acid here. Okay. Once that happens, okay, once we form our initiation complex, the next thing that we're going to do is we are going to bring in our large subunit, okay, that's going to bind onto this entire complex. Okay. The large subunit is going to contain 3 different compartments within it. Okay. The first compartment is called the A site. Okay. The A site is where any tRNA that is charged, which means it's a tRNA that's bound to an amino acid. Okay. An incoming tRNA is going to bind, or is going to income. Sorry, let me try that again. We're going to have an incoming tRNA molecule that is going to enter at the A site. Okay. So the A site is going to be where the tRNA is going to enter into this entire complex here. Okay. And again, it's going to be charged, meaning that it's going to already be bound to its amino acid. Okay. Once it enters the A site, so that's one side, that's the A site. Okay. The next site in the middle is called the P site here. The P site is where we are going to have a tRNA that's going to have a growing polypeptide. Okay. But that polypeptide has not dissociated, has not detached yet from that tRNA molecule. Okay. So it's still just residing in our P site. P site, you can kind of think of it as the growing polypeptide site. Okay. And then the last site is the E site. Okay. So once the polypeptide detaches from the tRNA, the tRNA is then going to enter the E site. The E site is going to be where the tRNA will exit from the ribosome from. Okay, so E for exit. Okay. So it's going to be uncharged at this point, okay, because it's not going to have its tRNA attached. Okay, it's going to be released so that it can then become recharged and bind onto another tRNA molecule later. Okay. So the A site is going to be where the tRNA is going to be coming in. P site is where our polypeptide is going to be growing from, and the E site is where we're going to eject our tRNA out of the ribosome from. Okay. All right. Now looking at the tRNA molecule, okay, each tRNA molecule is going to have what's called an anticodon. Okay. This anticodon is going to be complementary to the codon, okay, of the mRNA. Okay. So here's the anticodon of the tRNA. Here's the codon of the mRNA. The mRNA has a GCC, so the complementary anticodon would be CGG. Okay. And so this is what's going to allow the tRNA to bind onto the mRNA and then bring that amino acid into the picture. Okay. All right, so this is going to be how we're going to actually form our protein here. Okay. So the ribosome is going to be shifting 1 codon at a time. Okay, so every 3 nucleotides is going to be shifting from, okay, 1 codon at a time. Okay. What's going to be happening first is we're going to have our empty A site here, okay? And so we're going to have a tRNA molecule, a charged tRNA molecule, okay, with its amino acid attached, okay, that's going to enter the A site. Once it enters the A site, okay, it's going to add its polypeptide to the growing, it's going to add its amino acid to the growing polypeptide chain here. Yeah, there we go. Okay. So it's going to add its growing polypeptide to the new amino acid here. Okay. And so now the one in the tRNA that was in the P site is now uncharged. Okay, it does not have its amino acid attached anymore. Now that it's uncharged, it's going to move into the E site. Okay. Once it moves into the E site, that opens up space for the A site to take its spot, so the tRNA in the A spot to take its spot in the P site. Okay. Okay. Once it enters the P site, now it's chilling there. The one in the E site gets ejected out, and then you start this whole process all over again. Okay. So again, just to kind of recap, okay, we're going to have our tRNA that's bound to an amino acid. It's going to enter the A site. Once it enters the A site, the amino acid will then be attached to the growing polypeptide that's attached to the tRNA in the P site here. Okay. Once that attaches, the tRNA in the P site is going to leave, okay, and move into the E site and eventually get ejected out. Okay. The P site is now open. The tRNA in the A site will then move into the P site, take its spot, and then you start this whole cycle all over again here. Okay, so here's just kind of some more pictures, a larger picture to kind of help see this process a little bit better. Okay, so tRNA coming into the A site. Okay, once it comes in, it adds its amino acid to the growing polypeptide. Okay, the tRNA that was in the P site is now uncharged. Okay, it no longer has its tRNA here. It no longer has its amino acid. So what's going to happen is it's going to move into the E site, get ejected out. The one in the A site will then move into the P site, and then you restart this whole cycle all over again. Okay. Okay. Any questions on this process before we go forward here? So Kimberly's question is, does the amino acid in the A site ever have more than one polypeptide? No. So it only carries one single polypeptide. Each tRNA is only geared to hold a specific amino acid and only one. Okay. So for example, if we kind of go back to this amino acid, for example, this tRNA here is going to bind to the GCC. Okay. So it's always going to carry one specific amino acid. Okay. Whichever amino acid codes for, whichever amino acid is coded for by GCC codon. Okay. And it's only going to carry one at a time. Okay. So one at a time will be carried, or will be attached, okay, and linked every single time. Okay. Does that make sense? Hopefully that makes sense. Okay. Okay, cool. Okay, any other questions here? Okay, cool. All right, so question here. So we have our RNA sequence, AUG, CCG, AAA, CGU. Okay. So which of the following statements describes how the RNA sequence specifies the production of an amino acid chain? Okay. A, each set of 3 bases codes for a single amino acid. B, each group of 3 bases codes for an enzyme that binds amino acids together. C, each base produces electrical signals sent to the cytoplasm to initiate protein assembly. Or D, each base codes for 1 amino acid. Okay. How do we know how we're going to produce our amino acid chain here? What do we think? So I'm seeing B. B is not correct. So B's out. Okay. So which one is correct here? What do we think? Anyone have an idea? So Brianna's got this one right. So each set of 3 bases is going to code for a single amino acid. Remember, each of these 3 bases, A, U, G, okay, that's going to be 1 codon. And each codon is going to code for a single amino acid. Okay. Then you have the next 3 bases, C, C, G, that's going to code for a second amino acid. Okay. Then we have AAA, that's going to code for our third amino acid. Okay, that's going to be our third codon for our third amino acid there. Okay. So each set of 3 bases is going to code for a codon, and each codon is going to code for a single amino acid. Cool. Okay, so we'll go through this process here, and then we'll go and do a quick example. So here we have translation. So this is our last phase called termination. Okay, so termination is going to happen when the ribosome reaches one of these stop codons. Okay, either UAA, UGA, or UAG. Okay, when any one of those 3 codons is reached, what happens is the polypeptide ends up getting released. Okay, so here's our amino acid, or our amino acid chain, our polypeptide chain here. We hit our stop codon, and so what happens is we release that polypeptide into the cytoplasm. It eventually goes off and does its thing, okay? And then you start to break down the entire complex that you formed here. Okay? So the ribosomal subunits are going to separate from each other, okay? And then the mRNA is going to separate from the ribosomes as well, and then the mRNA is going to end up getting degraded. Okay? So once all that happens, your protein synthesis is now complete. So again, you're going to reach one of the stop codons, and then you're going to cause this entire situation to dissociate. Okay, ribosomes are going to separate from each other. The mRNA is going to separate from the ribosomes, and the mRNA will eventually get degraded down. Okay. Okay. So now we're going to go back and look at translation. So what we're going to do here, I believe this was it. Okay. Oh, is everyone able to see the activity again? Yeah, okay. All right, so what I'm going to do is kind of fast forward through this little video here. Okay. All right, so we're going to try translation here. Okay. And so remember, this video is a little bit kind of funky in that it skips the AUG, our start codon. Well, let's assume we've already hit our start codon. Okay. Our next one here is we have a GUC. Okay. So here we have a GUC. So where would we find the complementary anticodon on one of these tRNAs? Which one of these amino acids would carry that particular anticodon? CAG, perfect. So that would be our valine amino acid. Perfect. So we add our valine in. Okay. Then the next one is we have our CAG in our mRNA. So we need to find the tRNA that has the complementary anticodon. Okay. So which amino acid would that one be? That would be GUC. So that would be glutamine. Perfect. Okay. So glutamine becomes the second amino acid here. Okay. Now we have CCG. Okay. So which one has the complementary anticodon? GCC. Perfect. So that would be carrying proline. Perfect. Okay. Now we have CCA. So which one would carry the anticodon? That would be GGU. Perfect. So that would be proline. Okay. Now we have GCU in the mRNA. So which tRNA carries the corresponding anticodon? CGA. Perfect. Yeah. Okay, cool. Now we have UGU. UGU would be ACA. Perfect. That would bring us the amino acid cysteine. Perfect. Okay. Now we have CGA. So which anticodon would that one be? GCU, perfect. So that would give us arginine. Perfect. Now we have GAU, which should be what? CUA, perfect. Aspartic acid. And then we have UGC. Right, that would be ACG, which would be cysteine. Perfect. Here we have AUC. UAG, which is isoleucine. Perfect. CUA. GAU, leucine. Beautiful. Okay. So now we've formed our protein through translation, but there is a little piece that we're missing here. And we should eventually reach one of these stop codons. Okay, so let me rewind back a little bit here. So eventually, as we continue performing translation, we'll eventually reach one of these stop codons. So here we reach UAA. Okay, so this is one of our stop codons. Remember, the stop codon is not going to code for an amino acid. This just tells you when you're done with translation. Okay, and so at that point, what will happen is the protein that you just produced is going to detach from the mRNA, and the ribosome, the tRNA will go away, the ribosomes will dissociate, and the mRNA that you produced or that you were using is going to break down. Okay. Okay. So that is translation. So that said, there's a couple ways of determining. Is everyone able to see the PowerPoint again? There's a couple ways of determining which amino acid is supposed to be coded for. Number one is you can use that chart. Okay, the codon chart will tell you what mRNA you're looking for and its corresponding amino acid. Okay, or if you don't have availability to the chart, you might look at the tRNA and its corresponding anticodon. Okay, but in most cases, you'll usually have the chart, and that's how I'm going to be grading you going forward here. Okay? That's how we'll typically be seeing it. Okay. All right. So, question here. So we have an RNA sequence that is 12 bases long. What is the maximum number of amino acids that can be coded for by this sequence? Okay. Is it 12, 3, 4, 6, or 36? If we have an RNA sequence that's 12 bases long, what's the maximum number of amino acids we can have? Anyone know? So it's not E. E is not correct here. It's not 12 either. Yes, the answer is 4. Right. 4 amino acids are going to be coded for by our 12 RNA sequence long amino acid here. So let's just say we have, here's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 bases in our RNA sequence. We're not going to worry about the nucleotides. We'll just kind of do this for the sake of you being able to see it. Okay. So remember, in our RNA sequence, we're going to be breaking everything down into 3 nucleotide codons. Okay. So this would be 1 codon. okay, which would give us one amino acid. Okay, I'll just put AA for amino acid. Okay, then we have 1, 2, 3, giving us our second amino acid. Okay, then we have 1, 2, 3 nucleotides giving us our third amino acid, and then here's our last one. Okay, our last set of 3 giving us our fourth amino acid. Okay, so if you have an RNA sequence that's 12 bases long, okay, you're going to be able to form 4 amino acids, okay, because you're going to be taking it into in threes. Okay. So Kimberly's question is, what about the start and stop codons? Oh, okay, good question. So for you would have to take into account the start and stop codons as well. In this case, I was asking about the maximum number of amino acids. So here I would just be assuming maybe we're in the middle of our amino acid sequence. But if you were looking at the entirety of your strand here, oh, I guess I see what you're saying in terms of it could possibly be 3. Yeah, okay, I can see what you're saying. So let's say that this was our, how do I want to do this? For now, we're just ignore the stop codons for this particular scenario. Okay, let's just say we're looking at 12 bases inside of our RNA sequence. So from those 12 bases, we're going to be able to form 4 amino acids. Okay, but if we were to say, I think I see what you're saying. So if we were to say that our entire sequence was 3 bases, or it was 12 bases long, the last 3 would be the stop codons, and so you would really end up having only 3 amino acids. Okay, so very, very good kind of. Very good question here. Yeah. Okay. But let's just assume we're looking at the middle of our RNA sequence. Okay. That's that. All right. So now, let's say a deletion mutation has occurred. Okay. So we had a deletion mutation occur, resulting in an amino acid sequence that is only 11 bases long. Actually, this should be an RNA sequence. An RNA sequence that's only 11 bases long. Okay. So if our RNA sequence, actually, let me go ahead and fix that here. RNA. Whoa, sorry. 1 second here. RNA sequence. Okay. So now let me try this one more time. Okay. Okay. So now, ignore the big red box. Okay. We say a deletion mutation occurred, resulting in an RNA sequence that is 11 bases long. Okay. So again, let's assume we're looking at the middle 11 bases here. So what is the maximum number of amino acids that we can get through this sequence here? What do we think? Whoops. I don't know why my chat is not wanting to come up. We should be able to see that the answer should be 3 bases here. Okay. So again, because you have 11 bases, okay. If we were to draw this whole situation out here, oh, there we go. Okay, now it's about to show. Okay, yeah, so 3. So again, if you're looking at 3 bases, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, you want to form your 3 nucleotide codons. Okay, so here we'd have 1, here we'd have 2, here we'd have our third one. Okay, but we don't have enough to make our third codon, so we would stop here. Okay, and so this is kind of where we can see kind of the danger of having mutations. Okay, so if you have a mutation, that can cause some alterations inside the protein that you're able to produce, because you're going to be missing entire amino acids, and in some cases you may even be adding incorrect amino acids and causing a defective protein to be made. Okay. Perfect. Okay, so here's another question. Okay, so here we have an RNA sequence, ACG, AAA. My thing went away. GAU. So using the codon chart, what is the sequence of amino acids that is produced when this gene is translated. Okay? So these are your mRNA sequences here. Okay. These are your mRNA sequences, and then the corresponding amino acids. So what do we think our sequence of amino acids should be here? I'm going to take a minute to see if you can figure that one out. Yeah, so Kimberly's got this one right. So the answer would be A. Okay. So the way you would do this is you look at your first codon, ACG. Okay. Where do we see our first ACG? We see our ACG over here. So this would give us our amino acid threonine. Okay, so that'd be number 1. Then we look at the second one. We have AAA. Okay. We look for where we would find our AAA, and we find that over here. Okay, so that would give us lysine. Okay. So that would be the second amino acid here. Okay. And then we have GAU. Okay. So GAU would be right over here. That would give us aspartic acid. Okay. And so that would be our third amino acid here. Okay. And that's all you do. Okay. We look for each individual codon. Okay. You look for where you would find it on the chart, and then whatever corresponding amino acid that is going to be the sequence that they will show up in. So perfect. So A is absolutely correct. Threonine, lysine, and then aspartic acid. Cool. All right, and that is it. Are there any questions on how this entire process goes here? Any questions at all? Okay. If not, what I'll do is I'll show you guys what you'll be doing for your lab this week. And this week's lab is going to be pretty short. We're just going to be doing activity 2 inside the lab manual. I pretty much wrote everything out here, so you really don't need to do everything in your lab manual. You can just do it on Microsoft Word, and it's uploaded onto Canvas as well. Okay. And so the first thing I want you to do is go through. This is the website that we just used for our simulations for transcription and translation. There are some videos, so go through, watch the videos, and then if you want extra practice with going through transcription and translation, you can do that as well. Okay, that's going to be the first thing you're going to do. The second thing that you're going to do is you're going to perform transcription. Okay, so here's our DNA sequences. Okay, here's one strand of DNA listed in blue across the top. Okay, here's the other strand across the bottom. And so you're going to be producing an mRNA sequence specifically using the top strand as the template strand. Okay, so the top strand is going to be your template here, and you're going to be using that to help you identify what your mRNA template would be, okay, using transcription. Okay, so once you've performed transcription, okay, the next thing you're going to do is you're going to copy your mRNA sequence, add it into here. Okay, you're going to underline the first AUG, okay, wherever you find your start codon. Okay, underline that first AUG codon. Okay, that's going to be your start codon, and then you're going to begin translation from there. Okay, remember, translation begins at the start codon, regardless of whatever amino acids, whatever codons are in front of it. Okay, translation only begins at the start codon. Okay, so you're going to include what amino acids you would find. Okay, and there's a chart on the next page to help you identify which amino acids should be going where, and you're going to write them in the correct order. Okay. And that'll be your translation kind of simulation here. Okay. And then you'll just answer the following questions here. So the 4 asks you how many amino acids are in your protein, and then 5 asks you about the anticodon for tRNA. B asks you what the corresponding amino acid would be bringing, and then C, 6 and 7 are just other questions here. Okay. So this is a pretty short lab, shouldn't take you too long, maybe an hour at the most, I think. And again, you'll have this chart here available for you to use when you're actually producing your protein through this. Okay. And that is it. That's all you'll be doing. Okay. Any questions on what we'll be doing for a lab this week? Okay. All right. So if there's no questions, you guys are free to go. Yeah. So Brianna, don't do it in your lab manuals. Because there is an error in the lab manual in the DNA sequences, and it's going to throw everything off. So just use what I used on Microsoft Word.